Thank you, everybody, for coming to research conference today. We have a special speaker from SomaLogic who's going to tell us a little bit about SOMAmers. This is Steve Williams, and he got his MD training from Charing Cross and Westminster Medical School at the University of London. Subsequently did a PhD in medicine and physiology related to hypertension. And as he told us earlier, wasn't quite sure what he wanted to do, so he did 3 more years in residency in radiology training as a part of a bigger theme in biomarkers. He then moved to Pfizer in the UK, where he headed a lot of the biomarker work, subsequently came to the United States in Pfizer and headed a unit specifically designed around biomarker development and implementation in drug development. In 2009, he became the CMO at SomaLogic. And today, the topic of his discussion with us is going to be uncovering complex patterns of biology in health and disease with SOMAscan, a breakthrough proteomics platform. Welcome, Steve.
Thanks, Kristen. Hi, everybody. Thanks for the invitation. It's great to be here. And I believe we're going to be stuck here. So just to say who I work for. So, of course, you know, I'm a shareholder in SomaLogic, and I'm also on the board of a little diagnostics company that's making a test for appendicitis. So I'm going to tell you about a new approach to proteomics. Before I start and tell you what we've done, I really wanted to talk a bit more about what the problems have been. And I think the key problem here is that we don't actually know very much biology. My boss, Larry Gold, the founder of the company, likes to say we only know 5% of the biology. So if you use the existing literature to identify what it is that you want to measure, what's interesting, actually you're missing 95% of it. Well, why is that such a big obstacle to us? Well, the obstacle is really that if you could measure lots of stuff, you wouldn't need to know what was in the literature. You'd be able to actually find out what those signals were. But until now, if you wanted to measure many things, you actually had to compromise on the sensitivity of the technique. There was this trade-off between measuring lots of stuff or measuring small amounts of stuff at very high sensitivity. And I'm going to show you how we've at least partially overcome that problem with a technique that can measure both many things and cover this dynamic range issue, this sensitivity dynamic range problem of proteins where there are 8 to 10 logs of concentration. What you're looking at in this little screen is an illustration of that. It's actually a picture of proteins from kids with Duchenne's muscular dystrophy. And these are heat maps of proteins that are correlated with one another. As you can see in the kids on the right, this doesn't actually show up on the screen. Can you actually see the pointer or not? Is it invisible to you too? Okay. So I'll wave my arms. On the right of the screen, you see the kids with Duchenne's, those proteins. If you're a good guesser, you'd have been able to guess that 22 out of the 1129 you measured were the ones that were moving together across the disease spectrum, and that they weren't moving the same way in the kids on the left. But you'd have to be very lucky. The nice thing is, if you measure many things, you can find these mathematical correlations. The other thing, of course, is that any one of these pixels might be 8 logs more abundant than its neighbor. So you need a technique that can measure many things over a wide range. And this is our secret sauce. What you're looking at is an X-ray crystal structure of a somamer, a slow off-rate modified aptamer. And that's that brown wiggly worm you see over there on the left. It has a DNA backbone. It's about 40 bases long. And it's binding to a protein, which is the brown amorphous blob. And we've known for a long time that DNA itself, just naked DNA or RNA, has a charge and it has a shape, and you can use it as a binding reagent. And that was the original vision of SomaLogic 14 years ago, that it would make these binding reagents and we'd be able to pull proteins out and measure hundreds or thousands of things. But it didn't work. We got stuck measuring about 50 things because the binding of an unmodified piece of DNA or an RNA is not specific enough or strong enough to overcome this dynamic range problem where even if you're 99.9% specific, if there's a protein that's 10 billion times more abundant than the one that you're trying to measure, that will be the dominant thing that you measure. And we overcame that problem by modifying the DNA so we could get more shape and charge diversity. And that appeared to improve the binding characteristics. And this X-ray crystal structure shows you that in action, that the pink bases you see in the middle interacting with that cleft in the protein, those are the modifications we've introduced to the DNA. So the picture shows the explanation for the improved binding. We make these things one at a time. We start with just that protein, a human target protein properly folded, and we incubate it with 10 to the 15th random variations of somers, and we find the one that binds the best affinity and the slowest off rate, because we use both of those in the assay. And you can see a little zoom in of the shape-based interaction in that close-up on the side there. This is a pie chart of what's in the 1129, and people often ask us, well, how did you decide? And the very simple answer is we didn't. We just went after all the properly folded, highly purified human proteins we could get, because that's where we start making our reagents. And that's not completely random, because in order for a protein like that to be commercially available or available from collaborators, someone somewhere has to think it's interesting enough to have made it and purified it. So it's a little bit like a popularity contest. But the nice thing is, as you see from these pie charts, it doesn't really matter where the origin of the protein is or what biological process it's involved in. They're represented widely on the menu. And that's important, because if you're looking for that 95% of biology that we don't know yet, you actually need to be able to look in all of those different subspaces. This is the principle of the assay. If we have a bit more time at the end, I can show you a more detailed explanation. But this is how it works. You start on the left with the protein, N proteins, 5,000 of them maybe in a plasma sample. And so you start with a protein molecule counting problem, which is really hard. And what we do is we use these somers, the 1129 of them, to make complexes. And through a couple of immobilization steps, we end up with just the DNA. What we do in the middle here is to immobilize the complexes so we can wash away proteins that didn't get complex, didn't find a somer. And we can also wash away somers that didn't find a protein. So at the end of the assay, you're left with these molecules of different varieties. And the number of these DNA molecules at the end is proportional to the number of those protein molecules there were at the beginning. Counting DNA is easy. When you're doing hundreds or thousands, you want to use an Agilent scanner where you pre-print this slide with all the complements of the DNA that you had, that you knew you'd introduced into the assay. If you're only measuring a small number of analytes, of course, you could use qPCR or you could use Luminex beads or something like that. But when you're measuring thousands, you want this. So the way this works is we've pre-printed this slide with the complements of the 1129, and we know where they are on the slide because we ordered it. So the physical position of any spot on that slide corresponds to a particular sequence. And the intensity of the fluorescence of a spot on the slide corresponds to how many molecules of this DNA bound to it. So the position tells us the identity of the DNA molecule, which tells us the identity of the protein it was raised against, and the amount of fluorescence in the spot tells us how much protein there was. And we have standard curves for all of these proteins so that you can extrapolate back from a fluorescence to a concentration if you need to. So these are the characteristics of the assay. I'm not going to go through them all individually. What you'll notice is there are other technologies that can measure things at very low abundance, and there are other technologies that can measure many things and with good CVs, but this combination of attributes is pretty unique. Today, we've qualified the assay to operate well with the list of matrices you see on the left, and there are others that are still under research. What we like to do when we optimize a matrix is to match the dynamic range of the reagent to the physiologic concentration in that sample, in that type of sample. And of course, at the beginning, when we get a new kind of sample, we don't know what the protein concentration is for all of these things. We have to find that out. So that's what we do to get from the right to the left, is we find out which reagents are on the floor and which ones are on the ceiling, what dilution should we run it at, should we split the sample up into 2 or 3 different dilutions and split the mix of SOMAs up into 2 or 3 so that each one is able to actually detect something. Because although we get to 8 logs of dynamic range, we do that. Each individual reagent only has an average of about 4 logs. And so we get to the 8 logs by splitting the sample into 3 and doing it at different dilutions with different mixes. We've done a lot of samples so far in the 5 years since I have joined SomaLogic. When the assay has been working, we've run about 40,000 samples. But we're ramping up. This has kind of been a real exponential increase. We're now building the capacity, or we have built the capacity to run about 40,000 samples a year. And we've looked in lots of different places, and our clients, external academic collaborators and industry clients have looked in lots of different places for biological signals. And you'll see a list here of all the different areas where we've had success in identifying biomarkers. It seems as if it's nearly universal that we always find something. I'll show you about how we control for false discoveries in a moment, because that's where my mind would leap if I heard someone spouting on about how we always find something. All right, so I'm going to start with talking about false discoveries and the effects of transporting samples and handling samples. And I'm going to use this little study to illustrate the big hole that we fell into when we did our very first study. When the most powerful biological signal from a 1,300 sample study in lung cancer was the difference between NYU and Pittsburgh. So how do we overcome that problem? Well, what we're looking at here is an experiment in 4 people where we had 80 samples. And on the right, what you're looking at is each spot is 1 sample. And there's a principal components analysis, where a mathematical technique that pulls out the most important patterns. And what you're looking at is the first 2 patterns plotted against one another. So on the right, each spot is a sample, so there's 80 spots. And on the left, each spot is a protein, same technique, same positioning. So on the left, there's 1,000 proteins there in the version of the assay where we were looking at. So how do we explain the position of these little dots on the screen? Well, let's go to the right. We know that there are 4 people, and you can actually see that there are kind of 4 columns of spots here on the right, and with the eye of faith going from the different color-coded, 1, 2, 3, and 4. So there's something different about this column of spots from these 3. And you hypothesize here that these belong to 1 person and these belong to the 3 other people. And there's something in this left to right shift, which is about the people. What could that be? Well, let's look on the left to see what proteins are involved in that left to right shift. And you'll see that at least 2 of them, LH and FSH, are gender related. And that left to right shift is indeed gender related. So that's a biology axis. That pattern that was pulled out was a piece of biology. So that's real. What's the other axis? So what's causing this up and down shift? Well, the up and down, you can see, is driven by a lot of those red spots, those red proteins, which are intracellular neutrophil proteins. So what are they doing in the sample when they're supposed to be in neutrophils? Well, what they're doing in the sample is they're the result of cells dying as the sample was left on the bench. They consume glucose, they run out, some of them die, they dump their proteins into the sample. So that's what you're seeing. The second most powerful signal you're seeing in this sample is dying cells from being left out on the bench too long. And this issue was really dominant, we found, in our very first big discovery study. And initially we were saying, well, we're only going to have to do prospective studies where we really carefully control all of the parameters for collection. And we ended up not having to do that. Because what we did was to turn that signal, that cell lysis signal, into a vector. and we can actually, we have several different vectors for different processes now. So we created the conditions in the lab where we could identify little groups of proteins that we use as different colored canaries in the coal mine. So for any sample that comes in, we can actually say what's happened to it in the past. And you can do a few things about that. You can throw out some samples if they're terrible, or you can throw out some analytes if you say, well, in my collection, there's a lot of complement activation going on. I'm going to throw out these 50 proteins. I'm going to use the other 1,100. Or sometimes you can adjust for that effect mathematically and kind of correct it back to a virtual perfect sample. But understanding and managing this has turned out to be one of the most important things that we've done. I'm going to go through a couple of normal variations. This one is a genetic variation, and what you're looking at is a protein concentration graph of a single protein. So just pulling out one protein out of the 1129, saying what's the concentration in a population of nearly 1,000 people. And what you see is that the apparent concentration of this protein has 2, and maybe with the shoulder on the right, 3 peaks. What's happening with SIGLEC9 to cause a population distribution of 3 peaks? Well, you can see on the right here that there's a SNP involved in this protein. And if you inherited 2 A's, one from each parent, you have entirely unmodified protein. So all your protein is what we made the reagent to. And you have the highest apparent concentration there in that shoulder of the peak on the right. But if you got one allele with the SNP in it, your apparent concentration is shifted over a bit into that peak. And if you got 2 copies of modified protein, the peak is shifted lower still. Now, this is a kind of clinically useless observation, but I like to use it because it shows you the effect of modifying proteins on the SOMA-based signal. In this case, this is a kind of an inherited difference. But if there's a biological signal that modifies the protein at the site of binding of our reagent, that will be reflected as an apparent reduction in concentration. And if it becomes important to you to understand what the cause of that is, then you need Art, wherever he is, to use mass spec and say, was this really reduced concentration, or was it simply modified? Next little kind of vignette is we looked at the response to a meal, and really the point of this slide is not the different. These are not abundances, they're just relative changes in time after eating a high-fat meal. The point here is that these were the top-ranked proteins, that it took one little study with a meal to pull out the physiology that took a long time to discover. You get insulin and glucagon, insulin growth factor binding protein, and so on, and FGF19, which is an allele protein. I didn't create this graph knowing the physiology. This came out just mathematically. These are the top-ranked proteins. And that's always reassuring to say, can this technology find what I expect to find? Well, this would assert that yes. I'm going to give you a couple of examples of pharmacology. This one in lung dysplasia. This was a little study in about 28 or 29 people per group. Half of them got iloprost, which is a prostacyclin analog, and half of them got placebo. They were all smokers or ex-smokers with lung dysplasia. And what our collaborators at the VA in Denver were looking for was proteins which correlated with clinical response. And that's what you're looking at in the left-hand box up here. You're looking at this particular protein, this one protein. The change in the protein was correlated with the change in dysplasia over time. Whereas in the other groups, if you carried on smoking, it didn't correlate. Or if you didn't get the drug, it didn't correlate. But if you stopped smoking and you responded to the drug, that protein was correlated with the effect. And there were 14 proteins like that. And what was interesting was half of them were in the VEGF pathway. So this is suggestive that maybe there are other targets in the VEGF pathway that would help lung dysplasia. Maybe it's not just prostacyclin. Maybe there are some existing products that you could try. But the point here is that you could find mechanistic pharmacology. You could look for it was particularly correlated with clinical response. And you could interrogate, using some systems biology, what might be driving that response. Another little interesting study, this time for the World Anti-Doping Association, I don't think Lance Armstrong was in this study, but he might as well have been. What they're interested in, of course, is finding the pharmacodynamic effects of doping even when the original compound has disappeared or can't be measured. So they're looking for the host response, if you like, to doping. And the part of the host response they really want is that which persists longer than the doping period itself. And what they did here was to dope some people with growth hormone over time. And what you're looking at here in these little graphs is the time-related curves from time 0 on the left to 7 days, 14 days, and then the last column on the right is 1 week after dosing, after doping had finished. So what they were looking for was the proteins on the bottom, ones that kept on going after the doping had finished. What was actually interesting was this effect, this persistent effect after doping, was more common than any of the others. It really suggests that the pharmacodynamic duration of human growth hormone is predominantly this. And of course, that's why people use it to dope with. They don't use it because they feel a rush on the day they take it. They use it because it persists and it lasts for weeks. So maybe this shouldn't have been a surprise to me, but it was. I'm going to show you a little bit of tissue pathology because, of course, this can be applied to tissue. Looking in lung cancer tissue, you can investigate differences between adenocarcinoma and squamous, which is what you're looking at here. These 70 biopsies have also been characterized for other mutations and expression differences, which we are working on today. So today I can only show you the adeno versus squamous. What's interesting here is that squamous cell carcinoma looks proteomically a lot more active than adeno, because if you're trying to find adenocarcinoma proteins only, there aren't very many. And if you find proteins that change in both, there aren't any where the adeno, where squamous is less active than adeno. There are none where this pattern exists. But with the, when you do get proteins that are different in both types of cancer, squamous is always higher than adeno. And then there are a lot of proteins where it's squamous only. And I'm going to move on to actually finding the same kind of cancer in serum. Sorry, this doesn't show up very well, but what we did here was a study in about 363 samples. I told you a few minutes ago we did this big lung cancer study and the samples were all over the place. What we went back to do was to say, okay, what fraction of those samples are really well collected and don't have case control bias in, because that was the other nasty little secret we found in that study, that the cases and the controls were actually handled differently. And until you have the detection methods that can find those biases, they look like disease biology. But in fact, it was caused by differences in transport and handling of the cases and the controls. So once we'd extracted a nice set out, did discovery on that, and then we, in a kind of a distribution that mimics the lung cancer screening population, and then we did 2 independent validation sets on it. And the kind of thing you get is this spiky graph across the middle this is a graph of p-values, negative logs of the p-values. So a big red spike means a small p-value, an upregulated protein, and a big green spike means a small p-value, but a downregulated protein. And what I'm showing you here is 2 markers, both of which had pretty good p-values. On the left, one which is robust, because if you look in the controls only, and that's the population distribution of controls only across 4 different sites, they match. But the one on the right, HSP90, it's a nice cancer marker, but as you see in the controls across the sites, it's quite variable. And that's because there's HSP90 inside neutrophils. And so if you're not controlling the time on the bench very well, you get a lot of variation in this marker. And because we have a lot of choice, there are so many markers that are changed, we can keep markers that look like the one on the left, and we can ditch markers that look like the one on the right to get a more robust product. And these 3 ROC curves you're looking at, remember where you want to be, is highly sensitive and highly specific, which would be up in the top left-hand corner. We aren't quite there. But what you're looking at here is training and 2 different blinded validation sets. And the green is squamous cell, and the red is adenocarcinoma, and the black is the combination of the 2. So you see that tissue proteomics, where we found that squamous cell was proteomically more active, is also reflected in the serum and downstream manifestation in the serum. It's probably also more important to find squamous cell because the doubling times are shorter and it's more aggressive, and some of these adenocarcinomas that you find on CT screening are probably have doubling times in years. Now I'm going to move on to cardiovascular disease, and some of this is really breaking data. I was making some of these slides at the airport on our delayed flight yesterday. So I'm not sure I've even drunk in some of the full implications of what you'll look at, but I hope you'll find it's very exciting. But I'm going to start the story a little way back with a paper from Rich Lee and Amy Wagers at Harvard, and they published this paper in Cell where they used SOMAscan in mice. So remember I told you these are all made against human proteins, so what the heck are they doing in mice? Well, there's enough homology between the human and mouse proteins that are probably about a third of them work just the same as their human counterparts. And a third of the analytes measure something, but the dynamic range is different, and a third of them don't work at all. Nonetheless, they went searching using the human SomaScan for the explanation to this problem. In 2001, they'd published this study that showed if you take aging mice who get diastolic heart failure, rather like aging humans, and you connect a young mouse to an old mouse with heart failure, the old mouse gets phenotypically younger. So the question was, what is it? What's the transmissible agent that's doing this? They had tried lipidomics and metabolomics, had failed to find the explanation, and here's the list of different proteins that they found between the old and the young mouse. This one circled in red is the highest protein on the list that was deficient in the old mice. And what they did then was to get GDF11 and inject it into the old mice without connecting them to a young mouse. And it reversed the heart failure, which you see on the right, the heart weight to tibia length went down after injecting GDF11. And what you see on the left is the differences in GDF11 between old and young mice. So hold that thought because I'm going to come back to that. The New England Journal was kind enough to write about maybe the technology is enabling a new era of molecular humors, and I'm going to try and reinforce that message in the next few slides. So I'm going to move on to human cardiovascular disease, and I've tried to think here on what, if you could wish for everything, what would it be? Well, I want better prognoses than existing clinical risk scores or existing markers. I don't want the commonest presentation of coronary cardiovascular disease to be sudden death. But I'd like it to be sensitive to all events, not just MI, but let's dream here and let's say we want something that measures everything. As I said in lung cancer, I also want these things to be robust. I do not want them to be have to carefully and specially collect these samples in a different way to ordinary blood. I want them to resist the trials of collection and transport. And I want it to go across different populations, and I want it to change if risk changes. So the reason I hate some of the existing risk classifications that depend on age or gender or smoking history is they don't change. If you change your lifestyle and lose 100 pounds and go on an exercise program and you take your statins that you weren't taking before and everything, your risk has probably gone down, but your score may not have changed by much because it can be dominated by the fixed factors that can't respond. So I want responsiveness to change. And the rest we'll go on with. So we did a study to start this process, looking for prognostic proteins. And we did the first study in nearly 1,000 subjects from the Heart and Soul study, where they took a single blood sample and they followed these people up for 8 years. And these were high-risk individuals. They'd already had an event or a stent. So the event rate was pretty high. They were all pretty much on the ceiling for their framing and risk score. But nonetheless, they were followed up. And in fact, even though they're called high risk, most people didn't have another event over the 8 years. So yeah, they're higher risk than the average population, but if you were trying to manage medical resourcing, half of those people didn't need any special medical resourcing. We also ran another 1,000 samples from a cohort study in Norway, where we matched the entry criteria for the sample selection to Heart and Soul, and that's our validation study. So what I'm going to be showing you is proteins that were discovered on Heart and Soul, and then any models were fixed and they're going to be applied to Hunt. So the validation set, we have not done anything special with it. We haven't recalibrated the models. We haven't adjusted the proteins. We did no discovery on the validation set at all. And what you're looking at here is a list of 17 proteins that Lasso, a kind of mathematical feature selection technique, found to be of interest in heart and soul. The black lines on the right are the hazard ratios. So 1 is in the middle. 1 is here, a hazard ratio of 1, 2, and 4. So you see the ones up at the top right-hand corner have a hazard ratio of nearly 4. Those are the ones where an elevation in this protein is associated with greater hazard, a greater number of events per unit time. The ones on the left of the 1 into a quarter or an eighth, sorry, half, a quarter, and an eighth. Those proteins is where a deficiency is associated with greater hazard. That's why they're on the left. And what you might notice here is this is GDF11, independently discovered, deficient in 1,000 human subjects related to cardiovascular risk. The other thing you notice is that the red and the black bars, the black are from heart and soul discovery, and the red are just what was that protein doing in Hunt in the validation set. I think they're remarkable. There's a remarkable agreement. We lost some. One of the reasons we might lose some is that actually the blood samples in Hunt were left out on the bench for up to 24 hours. So what we're doing here is we're trying to find proteins which relate to hazard and which are robust to real-world variations in sample handling. So now we can make a model out of these and say, well, how does that do? We actually didn't use the information about which ones had fallen down in Hunt to make the model, so that's something we could do in the future, but again, as we only trained on the discovery set, we did not use any of the information on that to change the applications to the validation set. So this is a graph of survival curves over 5 years of a model that uses 11 of those proteins. So having used Lasso to find interesting proteins, we then did a Bayesian backward selection to find a model of 11 proteins that we then used to predict risk. And what you're looking at is quartiles, so quarter of the population each in the discovery study on the left. And each line is so roughly 250 or 224 subjects. And you'll see that if you had the worst score here, if you were in the bottom quartile for that, for risk based on proteins alone, fewer than half of those subjects survived 5 years without an event. Whereas if you were up in the top quartile, you probably only about 5% of those subjects had an event or died within 5 years. And then the other remarkable thing is that when you take that model and you don't recalibrate it, you don't do anything to it, and you apply it to the validation set, the survival curves in Hunt are nearly as good. The hazard ratio on the left is 10 and the hazard ratio on the right is 7. And that's despite the fact that, as I say, these samples were left out on the bench and we lost a couple of those proteins. Here's the ROC curves for 2 different years. What you're looking at here is the curves out here. The outer one is discovery on heart and soul. The inner one is validation on Hunt. And then the Framingham curves in discovery and validation are the 2 rather poor ones on the inside. Now, another question is, well, what happens if you use clinical data and the proteins? The answer is you don't get any better. One thing we did do was to say, if you take that 17-protein Lasso model and you put in the pot all the clinical data available, and then you do your Bayesian backward selection, what gets chosen? And the answer is only proteins. None of the clinical measures got chosen for the model, even when they were available to compete with the proteins on a level playing field. And it's quite interesting, really, because I think if you think of the clinical measures like age and gender and other stuff, they're surrogates for something else. They work epidemiologically, but they're not causal. They're not personalized. There's something that they reflect. And I guess what we're learning here is that actually proteins are closer to that causal pathway than some of those clinical features. This is one of the things I was putting in at the airport last night. what you're seeing here is the probability of an event, actual probability calculated by the Framingham score, and the probability of an event calculated by the protein score. And the red dots are the people who had events, and the green dots are the people who didn't. The assay noise and perfect unity is down the middle here. What you see is that the Framingham doesn't really discriminate between red and green. The other thing that's a surprise is there are some people up there with a probability of 1 of having an event from the protein model. And you say, well, why are some people way up there and some people down here? So if you take the red dots and say, let's plot them against time to event, you get this graph, which isn't showing up very well. What you're seeing here is the 11 protein model score against time to event. So these guys up here, they're in this group. They were having an event within the next year. And then as time went on, as you got closer to the line, it was actually correlated with a longer time to the event. So in a way, what we're seeing here is that the risk score from the proteins is correlated to the proximity of the event for that person. Whereas in contrast, the clinical score, the Framingham-based scores, is not. That's, again, not surprising, because I'm going to go back to what I said a minute ago. Well, those are epidemiologic measures. When you put age and smoking history and gender into an equation, that can't change with the proximity to an event, can it? Whereas the protein model can change. The other interesting thing is that if you instead of training a universal predictor, what happens if you train the protein model for just one event type? And you look at how are any of those proteins that are optimal for one event, are they shared with proteins that are optimal for another event? And the surprising answer is not much. The key takeaway of this rather hard to read Venn diagram is there's more high numbers around the edge. So there's more proteins which are really not shared, like out here, not shared with many other event types. There's not like 10 in the middle here which are common to all event types. Each event type has a slightly different signature. So this brings you to think about, well, you could have a universal predictor for overall probability of an event and time to an event, and underneath that you could have little panels of proteins that say, and the event is most likely to be an MI. So going down my wish list, where have we got to? Well, I think we've got to some prognostic performance. I know that we need to build the weight of evidence beyond, in this field, 2,000 subjects is nothing. We need to keep on building that weight of evidence. But it does look as if the prognostic performance in high-risk subjects is better than the things we have available to us today. It does look as if it's sensitive and it's robust. We've tried it in some different geographic populations, Norway versus the United States at least. And these things at the bottom we don't yet know. I'd say there's circumstantial evidence that it responds to change in risk because of that time to event. We're not sure about that. We also have some 5-year samples within Heart and Soul, and it does appear that if you look at the 2 different samples in subjects who are approaching an event, the score goes up within the subjects. But that's still a bit weak, and the other things we have yet to do. So this is what we're going on to do. We think that the work left is to build more of the same, but also we can actually do a new paradigm here. We haven't chosen a protein first and ask, what's it good for? We're doing it the other way around. We've got so many to choose from. There are about 120 proteins that were prognostic. So if we're trying to build the optimal product, we can actually put in all the combinations of proteins in panels and subpanels that do all of these jobs, that are prognostic, are robust, are responsive to change, do tell us about the time to event and the type of event. We're not stuck with a single marker that we have to say, what fraction of this does it do? We can design our panel to do all of these things. And you see there what the other things we're interested in doing. So I haven't got time today to talk about these other things in terms of outside cardiovascular disease, but we have done some quite substantial studies with blinded validation in kidney function, a more precise measure of GFR without having to inject dihexol. We also have a blood signature of liver fat that correlates to liver biopsies without having to do a biopsy, and fibrosis. And we also appear to be able to detect some diabetic complications in the blood. And then nutritionally, one of the interesting things in the nutrition study was and it was a study from subjects in the Netherlands, when they were given an anti-inflammatory, antioxidant diet with green tea and fish oil and yogurt and other stuff, vitamin E, the protein that changed the most with that diet was PCSK9, which I'm sure that a lot of the cardiovascular folks here know there are some PCSK9 antibodies in late development by several drug companies. And at least here it was shown that nutritional status can actually affect the circulating levels of PCSK9 without the need for antibodies. So you could incorporate that kind of thing, and we're working with Nestlé to identify more things like that, where elements of nutrition could contribute to an overall picture. And what do we want this for? Well, because I don't want to do one diagnostic test at a time anymore. Because we have this technology, which is a great discovery engine, wouldn't it be great if it could also be a clinical product where you measure 1,000 proteins in somebody and you do all of these things and more? And you even have some free real estate where the system can learn new patterns and improve upon the old ones. That's our ultimate vision, to actually help people like you make discoveries, but ultimately to also provide the same platform where it can be put into clinical practice. And we think that if the care teams knew for an individual person about how each of these things had changed since last time they saw the patient, it might actually help medical resourcing decisions, and ultimately you would start to learn which treatments were the best match for which subtype of biology that the person has, actually driving their risk rather than the kind of epidemiologic age measures. And I'm going to round up by saying the reason we're here is because this technology today only exists in Boulder, Colorado. And one of the things we're doing is to export that technology. And we're just starting. And we're looking for a small number of institutions that would take SOMAscan and operate it for their own interests. And I'm going to go through a little bit about why we're doing that and what we're looking for. So we have this service model, and we've done a lot. We've done 140 studies. We ran 10,000 samples for clients last year. But sending all the samples to Boulder and having to go through that process has some limitations. So what are we doing about that? Well, we had this agreement with Agilent. They're supporting us and working with us because Agilent make the slides in the slide reader that we're using to measure proteins. You guys already use it here to measure DNA, but we're kind of repurposing that same reader for a different utility. This is what I call the win-win space. We think that this is going to be great for both of us because you guys, if you go through with this, you're going to be the first or one of the first places in the world that gets this technology, and you're going to be able to get grants as a result of doing that. And you're going to be able to support your own discoveries. And you get to work with us. And of course, we would support you in doing this. And Chris Barker, who many of you know, is here, and he's over here. He lives here, and so he can help that to happen. And of course, we like it because places like Duke have a wonderful reputation, and of course, we hope this will make other people more interested in our technology. And we're going to be providing kits for money to enable discoveries to be made. It's going to be a lot less expensive than running the service. But also, we get revenues from ultimately deploying this technology. But we need this early access program. We need partnerships with people who will really collaborate with us because we need to learn together how to turn this into an industrial process. So what we're looking for is these things here, which I think I already went through, all of which, of course, Duke has in spades. And it doesn't actually require that much to put SOMAscan in an institution. You need somewhere to put it. You need some people who are clever, and we need to reach an agreement, which is pretty straightforward. So I'm going to finish up now by saying thank you. We've got some great academic collaborators in lung cancer, particularly Jill Siegfried, Harvey Pass, and York Miller. And then I mentioned Rich Lee and Amy Wagers. And then for cardiovascular risk, we're working intensely with Peter Gans at UCSF. And then the kidney data I didn't show you from the University of Malmo. And of course, thanks to the whole team at SomaLogic. I'll be open for questions.
Thank you, Steve. That was a terrific presentation, and I'm sure just looking out here at the audience, there's a number of folks who have ideas about how they might use such a platform, but also questions they might like to throw your way. So if you'll raise your hand, we'll bring a microphone around. Got one in the back.
So I have a question to your cardiovascular use of the markers, which is very interesting. I think that first the positive, very good that you use the development and validation cohort, which is essential in this setting. However, I was not persuaded by your conclusions that we have a protein-based model substantially better than the existing prediction models. You mentioned in passing that when you added it to clinical variables, you didn't achieve anything beyond. You've contrasted with the Framingham model, which was developed for a very different population in primary prevention and kind of taken out of a bench, whereas here you still optimized your proteins. We didn't have any signs of better causality. So how would you propose your model could be used? How do I know that these proteins are not just a sophisticated way to incorporate aging into the model?
And so the Framingham model that we did use was the secondary prevention model that was published by D'Agostino. So they did validate this particular combination of factors for secondary prevention. So it was a match for the population. It's not the same as the Framingham primary equation. It is slightly different, and it was developed to predict events over 4 years, which is why we only compared it over the 4-year time period rather than the 8. So the Framingham equation was the match for this population, and the second part is really about how do we know we're not better than age. Is that when we give the mathematics the choice of incorporating age or a protein, it doesn't choose age. So you know that it's just not mathematically as powerful as the combination of proteins it chooses to substitute for it. And the technique, the lasso technique, and then some of the bioinformatics guys can help me out here, it does penalize using more and more markers as a kind of built-in penalty. What you do is you build thousands of models. You train them on half the training set and you test them on the other half. And you start with a very high penalty for using markers, so it'll only choose one. And you say, how many times does that protein get chosen in the model? And as you relax the penalty, of course, you let more and more proteins in, or more clinical factors in. So it's a kind of it's a mathematical selection process that chooses factors or proteins that are based on their performance and incorporation into models. And age doesn't win. So you can force it to include age, but you don't get any more performance because that piece of biology is already in there. And if you force it to include age, it'll drop out the proteins that actually found the biology of age because age will imperfectly substitute for them. So that's where we are today. We have also built, aside from framing, and we did just say, well, what if you force it to be only clinical models, and then the AUC curve for that is about halfway in between Framingham and the proteins. But there what you've done is invented a new clinical risk model that has no provenance, and it's not, and the performance isn't as good anyway. So that's why it's a long answer to why I still don't like age. And age can't be responsive, so.
Michael, did you want to respond to that? You are a mathematician.
So delighted to hear you use the D'Agostino secondary prevention model. Few people use it, and that's the appropriate one. now, I still disagree or have a problem with two other arguments. One is on the age level, the fact that one is selected in a stepwise or any penalized over the other might mean a minimal improvement. So to making a clinical claim to use a protein versus age, you need more than a minimal improvement. You need things that you mentioned like causality, alteration with statins. I totally agree with the fact that we can't alter age, and if we can alter the protein and show causality, that can be that can give us some potential, but we're not there. And the other issue is yes, age is in the model, is fixed, but we need to have a strong proof basically to eliminate it, and that is an open and interesting area, whether the model should be age specific or assume we have the wide age range and so on.
Thanks, Michael. We'll have some more time to talk about this right after research conference.
So I was curious how so you showed the one example where you had a natural variant where the SOMAmer didn't recognize the two of them equally well. And given, when you look at the crystal structure of these things, they're varying 1,000, 1,200 or so square angstroms on that average. So how common is that? I would think that would be a common issue, that you would have these natural variants in the population. So have you seen that in more than that one case?
So far, we've found about 61 proteins that have a multimodal expression profile like that. But I presume, and that's when we've only actually looked. We haven't had many samples that have been genotyped, and the sample size was pretty small. So the study I'm talking about that found 61 was only 90 people. So we're only actually finding the commonest variations. But I think to see a change like that, if you know. Yeah, so that's where we are. I think there are probably many more, but 61 is the number that we're at in a relatively probably underpowered discovery of that kind of effect.
Thanks. Nice talk. I was wondering how you deal with. Is this?
Can you hear Jeff?
Anybody hear me? Yeah. Okay. How do you account for any post-translational modifications? It seems like some of the probes that you're using, the aptamers that you're using, might not bind in the setting of glycosylation, myrcillation, or a number of others. So,
yes.
How do you account for that? Which obviously can occur in the setting of a lot of comorbidities like diabetes and other lipidoses.
So the question, if you didn't hear it at the back, was how do we account for post-translational modifications? A simple answer is that we don't. That if the modification changes the shape or charge of the epitope we bind to, it'll be reflected as an apparent reduction in concentration. And if it doesn't, if it's a glycosylation away from that site, it'll be invisible. And we actually don't know. If it becomes important, let's say that you look at those deficiencies in cardiovascular disease, if it becomes important to explain the apparent reduction in concentration because you really want to know the biological cause, then you go to mass spec and you get some samples of apparently high and apparently low concentrations and you say, is it really low or is it modified? We can't discriminate. We can make reagents that discriminate, but what you needed to make those reagents is all of the different modified versions of the protein in advance, and then you'd have to do negative selection against each one and each combination. That can be done, but you have to have a reason to go after that. What we're doing is we're equipping people with a discovery engine that can expose some biological effects, that then there'll be times when you need to do more work to actually uncover what's at the root of those things.
So the way that you're discovering these things, you end up with multiple or potentially multiple SOMAmers per protein, and you're choosing the best one and you're going forward with that. You would sort of automatically get, or potentially automatically get markers for these post-translation modifications if you just included, say, the top 5 or the top 10. And there's certainly room on a microarray for more than 1,000 spots. So why the decision not to do that?
It's a good question, and when we do the selection of the reagents, we end up with pools that might have hundreds of different representations in them. Because we sequence them, you can see that some of them are nearly identical, and they're probably recognizing the same epitope. And there are some that look quite different, and they're probably binding to different places on the protein. And in fact, we've experimented with pulling somomers out of the pool to see if we can make sandwiches, and it seems as if we can find reagents that would make a sandwich assay possible. So you're right, they exist in the pools. Why we haven't included multiple members of the same class, I don't know. It's probably a kind of dimensionality problem in that you're actually creating 5 times as many dimensions that you're going to have to control for the false discovery rate of, but you're probably not getting 5 times as much information out. But I think we will go there. There are about 12 proteins on the menu that do have more than one somomer to the same protein, and I do think it's somewhere that will go eventually, but we haven't done it yet, and I haven't got a good answer for exactly why not, other than dimensionality.
So I think we have time for one last question. I'm going to give that to Dr. Peterson.
So that was a great presentation. I'm just curious now about the model you've gone to, the business model. I think it's rather unique, getting it out of the hands of an individual company that's had it for a long time and has been looking for the magic use, and now getting it to a lot of individuals. The question is, how do you deal with the intellectual property? Are you just deciding to make it on the idea that if somebody comes up with a magic test or a use case for this, that your product will be used everywhere and therefore that'll be thumbs up? Or are you trying to hold to the IP that might be around the actual test that's developed? Because both will have some value, right? I'm just curious which model you're going to.
It's the former, and that the discoveries you make on this platform are yours to protect in any way you want. What we want to do ultimately is to have, an FDA-approved platform that people can bring those small panels to, and we make that path towards commercialization easy. Today we haven't got that, but we are following the kind of the Illumina model of enabling people to make the discoveries they want to do, and kind of fueling open source discovery, and hoping that some subset of those will come back when we make that path to diagnostic commercialization easy.
So thank you everyone for attending Research Conference. I'd like to thank Steve for an excellent talk and the opportunity to hear about what SomaLogic's doing and the SOMAverse. Thank you.
Thank you.